Well, hello, folks. Welcome to this live AMA from Timeline headquarters here in Lausanne, Switzerland. I'm joined today by Dr. Dominique Denk, who's the principal investigator of the MitoImmune randomized clinical trial. And this goal of this AMA is really to share these really exciting results on the postbiotic MitoPure Urolithin A, but also for you to field questions to us, to Dominique. We'll just give a few minutes more, maybe another minute for folks to join in, and then we'll get started. There seems to be some echo. Do you hear echo? Okay. Well, it seems to be only, let's see if we got, let's see what's going on here. We have folks starting to join in. Okay, so, well, maybe I introduce you while the folks are joining, Dominique. So we have the pleasure to welcome Dr. Denk. Dr. Denk is a physician and tumor immunologist, and his research over the last many number of years has been at the interface of cancer biology, immune metabolism, and of course, immunology. Dominique got his MD from the Goethe University in Frankfurt, and has done 2 postdocs at the Frankfurt Cancer Institute, and is currently at the University of California, San Diego. With that, Dominique, maybe we kick it off. And the first question I always ask is, how did you get into immunology, and what brought you where you are? We know immunology is a very interesting and exciting field, and just one of the big findings just won the Nobel Prize for T regulatory cells. What got you into immunology and tumor biology?
Well, thanks, Anurag, and thanks for having me, obviously. So I think the answer is almost always the same when you ask a scientist. It's most often the case that it's by chance. And it was the same way for me as well. So I went to medical school, you learn about the body, obviously. At some point during biochemistry classes, you talk about the T cell receptor and you talk about the immune system and how all these different populations interact. And I found this very interesting, this complexity of the immune system, which then kind of turns into syntacticity, that in the end, all these complex populations, they work together for one unifying goal. And that I found very interesting. And when you look at most disease that we treated these days, they all come down to some sort of immune deficiency or the immune system interacting with the body. So it's almost unavoidable. And that's what really got me into studying the immune system. And no matter what I study, even if my projects start off without the immune system in mind in the beginning, they end up having the immune system involved in one way or another. So I think it's highly fascinating.
That's great. And that's pretty much a similar trajectory I took a number of years back, 20 years back. I was actually reading a paper that came out, and this was a longitudinal study in thousands of long living individuals, and they found actually that 2 organ systems, the immune system and the brain, are actually the 2 key organs of longevity. So maybe before we get into the study, for the audience here, would you mind like a quick 20-second primer on what is the immune system? You know, we all talk about the immune system as if it's one system or one cell type. Maybe you talk about, you know, what is innate versus adaptive, and maybe give us another 30 seconds pitch or sort of describe what is immune aging in general.
Perfect. Yeah, great question. So it all comes down to development of the body and what we're built to adapt for. So basically, the immune system is the body's police. Those are cells that patrol the body and that make sure that we don't have any issues with pathogens, that is, infections, cancer, for example. Without the immune system, we'd be exposed to bacteria, viruses, and we wouldn't be able to fight those. So this is pretty much what the body developed to protect us from our environment, to make sure we survive. And there's 2 levels to the immune system, which we as immune experts talk about. Number 1 is the innate immune system, which is in almost some sort of way more rudimentary trait of the immune system. That is a very fast-acting path that defends against bacteria. It's not very specific, but it reacts broadly to all sorts of things. If you cut your finger, bacteria come in, those get recognized and those get defended against very quickly. That's the one arm. The second arm is the adaptive immune system, which adapts throughout lifetime to certain challenges. It's not as fast, but it's way more specific. So, for example, if you're exposed to a virus, that being the flu, for example, most of the time you get a little bit sick, obviously. You'll be sick for a few days, but your immune system will kick in, it will fight that infection. And most importantly, your body is able to form a memory against that pathogen. It's able to then, once you re-encounter that pathogen, so once you face somebody that has the same virus 2 weeks down the road, you will not get sick again. So that's kind of the way the body adapts to certain challenges throughout lifetime. This helps us pretty much to be able to survive in the environment we live in. So that's immediate.
Yeah. And then what happens with the aging process? Do these systems go weak, or if you could lay out in the next 30 seconds, what exactly is immune aging and what happens there?
So immune aging is pretty much a maladaptive process. Our body, as I told you, changes throughout lifetime, it adapts to certain challenges. It adapts to all these viruses we encounter. So that's in some way a physiological process. But by this specialization, we lose a ton of potential to fight other pathogens and fight other challenges throughout lifetime. So what happens during immune aging is that the cells that are actually supposed to recognize new challenges, so-called naive cells, they decline. They go down and down and down as we get older. And this is probably one of the reasons why we're not able to fight, for example, novel infections. We respond less to vaccines as we get older. So immune aging, in a nutshell, means our immune function declines. It gets less malleable and less adaptive throughout lifetime.
And so I hear, this is great. So in one end, you're saying there's sort of an immunosenescence where these youthful stem cell like naive T cells that are like almost like the elite forces of policing in the immune system, they go away or they decline. Then there's this inflammaging idea that our bodies just accumulate inflammation over time. And then what you are actually have spent a lot of effort or time in the last 5-10 years studying is the critical role of mitochondrial health. Do you want to talk about how key mitochondria are, or how good your mitochondrial health is as a bridge between good immune health and poor immune health?
Oh, definitely. That's a very exciting and new field that people have been studying the past few years. So we all know mitochondria as the powerhouse of the cell, right? That goes to muscle, that goes to all sort of cells. And it's the same in the immune system, that our immune system needs you for mitochondria to act, and it needs you for mitochondria to act for a long time. So what people have found in the past few years, only 4 or 5 years back now, is that as we get older, our immune system accumulates mitochondria that don't work well. We call those dysfunctional mitochondria. And those actually reprogram the immune cells in a way that they're constantly activated in a low grade, but they are active and they keep promoting inflammation inside the body. And this means that as the body's immune system is more active, we have this inflammation, but our immune system is not able to fight acute challenges. And that's kind of this reprogramming of the immune system that helps. So long story short, we need good mitochondria in our immune system for the immune system to work and to help against aging, not just in the immune system, but aging of the entire body.
And that's such a great point you make. I think a lot of scientists, clinicians, and just the general community is now really waking up to the key role of mitochondria in different organs, but for immune health in general. So, what got you interested in urolithin A? And just for the audience, urolithin A or urolithin A mitopure is this gut metabolite postbiotic that has been in the past shown to sort of rejuvenate the mitochondrial health via pathway called mitophagy, which means recycling bad mitochondria into healthy ones. So what got you interested, Dominic, to study urolithin A?
It's a bit of a longer story, but I'll try to keep it brief. So
Sure.
It was by chance. About 4 or 5 years back at that time, we were studying the role of cancer, the immune system in cancer.
Mm-hmm.
And our indicated that if you induce mitophagy inside the body as a whole, this promotes some sort of response against the tumor, and tumors don't grow as fast and they become smaller even. So back at that time, we just wanted to confirm these findings. We wanted to use some sort of compound that induces mitophagy directly. So we reached out to you guys. We figured, well, urolithin A might be something to study. And back then we only had the cancer cell in mind. We thought we changed the cancer cell. And we gave Urolithin A to preclinical models, and it worked. We saw tumors were smaller, but we were very interested then to see that it didn't necessarily act on the tumor cells, but it was the immune cells that were completely reprogrammed. And the immune response was very important for this Urolithin A effect. And that's when it really kicked off, because we saw, okay, our immune system, most importantly, the cytotoxic T cells, the cells that actually fight cancer, they're deeply reprogrammed, they're way more active. And then we linked this to findings in the literature that showed us that as we get older, this dysfunction that we see in cancer is pretty much the same during aging. It's similar pathways, and T cell dysfunction and mitochondrial dysfunction is pretty much the same. And that's when we made the connection that, well, we have these findings in cancer, and we know this works specifically on the immune cells. What if we take these findings and put it into, let's say, healthy people, where similar pathways are affected? And this is how we really kicked off our studies and went from a preclinical models, from cancer to humans to aging. Now, of course, you want to bridge it back to cancer patients, of course. But
Sure.
very great story, I think.
That's a great summary. And for folks to, who are joining in, feel free to put questions, and we'll try to take them towards the sort of the last 5 minutes of the AMA. And also for everybody to realize that research doesn't happen overnight, as Dominique is saying. It took 5 years from a first idea. Oh, Urolithin A mitopure is known to boost mitophagy, and it was shown in many preclinical and many randomized clinical trials to have an effect on muscle. But to really take that idea forward into asking the question, does it actually do the same in immune cells, and then go to a randomized trial, takes about 5+ years. So let's get into the actual star of the story, which is you and your clinical findings of this randomized clinical trial. Help the audience understand how you did this, raising the bar to really do this randomized clinical trial for a supplement to look at immune aging, and what did the trial show?
For sure. So of course you can show many things in the lab, right? In preclinical models, in mice, you see news every day. So of course we saw an effect on Urolithin A in our models, but the next step, and that's what you really have to do, is to show, does this work in humans? And for us, it was very clear that we know in our models Urolithin A changes T cells. We wanted to show this in humans as well. And this is how we came up to designing this trial. We knew the elderly or middle-aged adults even have change in the immune system. We hypothesized that if we target mitochondria directly with Urolithin A, we can change these advantageous changes. And this is then how we designed the trial, that we took healthy middle-aged adults, no prior conditions, between 45 and 70 years of age, and we profiled their immune system to see what does their immune system look like, and we randomized them into Urolithin A or placebo, completely blind, double blind. We didn't know what the people were getting. They didn't know what they were getting. And then after 4 weeks' time, we would look at their immune system again and see, are there any changes? If you give a placebo to somebody, be it a sugar pill or whatever, nothing should change, right? This is the idea. Our immune system is somewhat constant. What we found is that if you give Urolithin A to people, or they take Urolithin A for 1 month, there were actually changes in the immune system. They had an increase of these useful cells, these naive-like T cells, which are capable of reconstituting the entire immune system. And that was very exciting to us, because in the end, this confirmed our preclinical findings that Urolithin A acts on the immune system. It does so in humans even, in a very fast manner, within 1 month. And apart from that, we could even show that when you take immune cells out of the body from these people, you re-stimulate them in the lab, you do all sorts of assays, expose them to bacteria, for example, they work much better. They're way more active. They're way more ready to clear bacteria, for example. So this is the gold standard pretty much in what you want to do in research. You want to make robust findings in the lab. You want to find the biology, and then you want to test that biology in a controlled setting in humans to confirm what you've actually seen without making any claims, obviously.
Yeah, fascinating data, and to see it in 4 weeks is pretty spectacular. The question I get, again, also as a physician and immunologist, is, well, doc, I'm taking vitamin C and zinc, and that's supposed to help my immune system. How different are these findings in terms of what we know about how vitamin C works, or or even other longevity, for example, nutrients like rapamycin gets talked about a lot, or NAD modulators get talked about a lot. How can you put it in perspective for the audience here? How different are these findings from just like say, we're right now in a seasonal flu November setting. How different is this from just popping vitamin C?
Okay. Short answer and long answer, I guess. The short answer is Urolithin A works completely different. What we show is that it actually reprograms the immune system from what I talked about, the innate immune system to the adaptive immune system. We see these cells have different genes, they act differently, they look differently, they function differently. That's a deep programming. What vitamin C and zinc, for example, do is a bit different. Those compounds, vitamin C, for example, is a coenzyme. That means certain functions inside cells need vitamin C to function. And there's actually decent amount of data out there showing that if people that have vitamin C deficiency, if they take vitamin C, that helps their immune system. So it's a fuel, and it only, at least what the data shows, only works if you have a deficiency. So you refuel something that's not there. This goes with zinc. Zinc, for example, is found in many proteins inside the body. Zinc can actually inhibit viral replication, but there's only any use to zinc if you have a zinc deficiency, which has also been shown in trials. So it's fuel pretty much. Zinc and vitamin C are fuel if you don't have enough inside your body. That's the only time it really makes sense. What we've shown is that Urolithin A is taken by people that don't have any sort of deficiency, and actually it reprograms the cells. So you don't restock something that's missing. You add something on top to what probably wasn't missing, or maybe called mitochondrial deficiency. I don't know, but I think it's a completely novel approach. And the data that we've shown, I think, is pretty solid that you can broadly apply this probably to people. We have to see, of course, for how long these effects last, what the real world outcomes are, but it's highly encouraging for sure.
Thanks for that great explanation on the key data. So let's take a few questions from the audience. The first question I see, one of the first many questions that we're getting is, so this could be like an immune modulator. Would you agree or not agree? And would this have a role for example, for autoimmune conditions?
Part one, immune modulator, I definitely agree. We deeply profile the cells, which means we take cells, we look at the complete genetic programming, we see they're modulated, they are reprogrammed, they're completely different than before. So Urolithin A, yes, is an immune modulator. For any use in certain conditions, we don't have the data. I think there's no way right now to do any intake advice or medical advice on this. We need to study this. The data is encouraging. What we see is that it's completely safe and tolerable for intake in our study and also previous studies that you guys have done. So it's super encouraging. And now we just have to move from what we've seen in healthy adults into actual disease populations, which, of course, it's a long, tedious process, but I think this is the only way to do this correctly. And the more patient you are in these sort of steps, the more rewarding this is going to be. We're not looking to make big headlines and say, oh, this works for X disease. We're going to do the long path, and it's going to be very rewarding and very robust data for people to look at.
Absolutely agree. I mean, I was trained by my professor many years back, and he always said research is two words: re and search. You always have to keep doing it over and over again to unlock, bigger things what a molecule or what a finding can do. Let's take a few more questions. There are questions about, can I take it? I have high inflammation, for example. Would this make sense to take it if I have high levels of inflammation? Whether I'm looking at, for example, C-reactive protein levels with my doctor, or for example, would you recommend this if somebody has high inflammation to take this?
So we didn't study this directly, but there's plenty of data from you guys also that shows that the intake of Urolithin A actually lowers C-reactive protein levels.
Yeah.
And our data actually indicates also that this constant inflammatory reprogramming of immune cells is abolished by Urolithin A. Yeah. So from this angle, yes, there's very strong data that Urolithin A actually lowers constant low-key inflammation.
Okay.
I don't know yet, but I think it's very encouraging and something to check out.
There's at least 3 or 4 questions I see coming on fasting is known to also have an effect on the immune system and on similar sort of things. Would this work if I did intermittent fasting? And I'll let Dominique answer, but the research we have done in the past, and of course we have not done a randomized clinical trial yet combining intermittent fasting with, for example, urolithin A supplementation, but the research done at a preclinical level, for example, shows similar biology being hidden, at least in species like worms, for example, there was an additive effect. So, but Dominique, do you think as a clinician, as an immunologist, there would be an additive effect on combining it with intermittent fasting?
Potentially, I would say. The thing with intermittent fasting is that there's tons of data out there for longevity, health span, that it has certain beneficial effects. We just don't know how it works on immune health. There's not real good data out there, and it's very hard to study because the questions, of course, how long do you fast? What is the calorie restriction, for example? So this is all hypothetical ideas, but the pathways that are being targeted are similar, I assume. If you can add up on top of this, or if Urolithin A even completely uses up all these pathways, we don't know that. That's a big study for sure. But I think the intake of Urolithin A would be a bit easier than fasting, at least.
And one of the more fascinating things I saw in the publication now, it's always amazing how almost, these all these Nature, and of course you've published in a very prestigious journal, Nature Aging. They have the key figures from figure 1 to 5 or 6, but a lot of the key data is in the supplementary, and I saw all the data on single cell level. So maybe you want to talk about the findings you saw at a single immune cell level. This is pretty revolutionary findings what you're doing in the study.
Yeah, definitely. So as a quick explanation, what is single cell sequencing? So what you do is you take, at least in our case, you take the immune cells from a patient and you send them through a machine where every single cell by itself gets looked at in very detail, which sort of genes are inside that cell. And genes kind of show us what kind of programs are activated and what that functional state of that cell might be. And that was kind of the idea for us to look beyond T cells, to look at the entire immune system. And what we found in this part of the study is that urolithin A targets probably the entire immune system in similar pathways. We see mitochondrial pathways being upregulated. And most importantly, we see all these programs that are associated with inflammation, they all go down across the board in the innate immune system, in the adaptive immune system. And this tells us that this whole idea of mitochondrial health, it encompasses the entire immune system pretty much, and it's quite a preserved pathway. And there's great potential for urolithin A to hit these pathways.
Great.
Of course, there's a bit hidden in the supplementary figures, but that's just the design of the study. You design the study in the first place, you tell people what you are going to study, and the study gets judged on these endpoints. And these are the endpoints which are in the primary figures, obviously. What goes beyond that and what's more exploratory, at least to us, is more interesting, but that we put in supplementary.
A lot of questions on combining it with NAD or creatine or other mitochondrial boosters. Any thought on that, Dominique? And I can share my thought in follow up.
What you would call mitochondrial boosters and all these compounds, which are pretty hot right now in the longevity space, there's not good data out there for immune health or even intake. In the case for NAD, for example, it's a coenzyme also. It helps reactions in the cell to function. It's not really clear how to supplement NAD. So NAD is not taken up directly by the cells. You have to take the precursors. We know that those might be tolerable, but there haven't been really extensive studies on that as well. And we have no idea how it works in immune health. So I think it could be encouraging to study that in the future, but that data is way more immature than what we have at this point. So
yeah, thanks, Dominique. There are almost many 3 or 4 questions about timing of intake and the study and what we see or recommend. I can chime in because we've done a lot of clinical trials in the past. Most of the, we've compared with or without a meal. So when first thing in the morning, when you wake up and almost a fasted stomach, you take it, or if you take it with a breakfast or any time of the day, in fact, the kinetics of absorption of urolithin A, whether you're taking the powder or the pill or the gummies, it's pretty much the same across the board. But as far as the randomized clinical trials, and that's where we kind of base our recommendation, and Dominique can chime in, we kind of recommend taking it first thing in the morning and an empty stomach. But Feel free to chime in on the timing of intake used in the study, Dominic.
For sure. The recommendation, the official recommendation was first thing in the morning, a glass of water before the meal. I know for a fact people didn't do that, but all of those, that's just the real world effect of how things go. And we saw the effects across the board. So I'm pretty sure that the timing does not necessarily matter. And it would be worth the study, but I think it doesn't matter. I think you guys from your muscle studies, I think you have bigger studies even. I don't think people completely held to that program of
yeah. There's a great comment saying I've been taking MitoPure I'm 51. I've been taking and in addition to the muscle effects, I really feel an immune boost while others around me were sick. I seem to stay healthy. Now, do you think that to have sort of a health span or a longevity effect, you need to hit on multiple organs, Dominic, like muscle and immune?
Oh, for sure, for sure. Aging is multifactorial, right? You have the frailties, so the muscle gets weaker, people fall. You have the immune deficiency if you get sick. So when you ask people what are they afraid of in aging, they don't say getting old, they say being sick. And that encompasses many different organs. So I think hitting the immune system is one pillar, obviously, but you need to hit everything else as well. And that is muscle health, that is brain health, staying fit cognitively. If we can hit all of these pathways with one or a few molecules in the future, I don't know, there's encouraging data, including Urolithin A, but it's probably a multifactorial thing that we will identify or refine our data on existing molecules and then combine them with actual lifestyle advice. Get enough sleep, take walks, be an active person. But yeah, aging is not a monofactorial disease.
So I see further questions on will I break my fast if I take it? So the pills that have 250 milligram per capsule and two that we recommend to take at a 500 milligram dose, they will not break your fast. They have zero caloric value. But if you do take the powders and the you will have a little caloric intake. So the pills will not break the fast for sure. Maybe as maybe we have 3-5 minutes left in this AMA. We take kind of a crystal ball. If I had to look at the crystal ball, so this first study is for 4 weeks, right? Where you see deep immune remodeling or immune rejuvenation effects on immune aging. What do you see as the sort of the next step? Where do you see the research in terms of Urolithin A and immune aging headed? If you had carte blanche and you had to map out the next study.
Yeah, I have many ideas, obviously, which I think we'll be able to link up on eventually. So one thing I've been emphasizing is the real world outcomes, right? So what does reprogramming of the immune system mean?
Yeah.
Does that actually mean I'll be more healthy? I'll have less flus, for example. I think this is the first thing to address in the near future, is to see that if supplementation with Urolithin A leads to less infections as an observational setting, and if it leads to response to vaccines, a better antibody production, for example, which you can do in a relatively quick manner. This will help us to actually know what does it mean in real world terms. If I had to look into a crystal ball more further towards the future in an ideal setting, as you said, I'm a cancer immunologist, and this whole story started with cancer, obviously. Our goal is still to bring this into cancer patients. I want to see what it does, of course, in combination with immunotherapy. And it's important to note here that immunity and immune system function does not necessarily happen in the blood where we measure it. It happens locally. It happens in the tissues. It happens where the cancer sits. It happens where the organ is actually infected. So I want to look actually inside those organs, inside those tissues. After Urolithin A intake, do we see that reprogramming inside the tissue as well? So if a patient has cancer and they get immunotherapy and they take Urolithin A, do we see these useful T cells also inside the cancer? Do they have different function? Do they interact differently? It's very, very high level study with lots of modern technologies, which is doable, of course. I think this should be one of maybe the second step from now to look at. But I think we should move beyond profiling towards actual real world outcomes.
Yeah. And so I hear your feedback on the real world outcomes, and I do agree that the next step would be to sort of repeat the study in conjunction with immunotherapy, which is where we are headed with your mentor, actually, Professor Florian Gretton, combining it with immunotherapy. But I actually think, if I think of the immune aging landscape, this was relatively, 45 to 7, or the median age was around 50, 55 years. Uh, If you had to redo this again, would you do it in older adults where the immune aging is even more enhanced? And would you, for example, do it before the flu vaccination, for example? Would that be a study design that you think could work to show effects on, even, I mean, kind of real world outcomes, right?
Um, I would not redo the study, actually,
Okay.
because there's plenty of data that immune aging is not necessarily linked to biological aging. Our immune system declines much earlier. When we talk about the aged people, you think 60, 70, depending on who you ask, of course, that number gets pushed deeper and deeper, obviously. So our immune system declines starting much earlier, starting 40 years of age, for example. So 45 to 65, I think we studied, that's an adequate endpoint, I think. And that's the number one step to see, are there actually changes in the profile of the immune cells? I think that's the first step without overrushing things. For a second study, of course, this is what you mentioned. I would take elderly people, I would give them a flu shot, for example, and then check after a few days what's the antibody production, and then check after a few months, do they have a change in disease outcomes? Do they get sick more or less? If they're sick, how long are they sick? How high are the titers in the blood? Those sort of things. And that's a very clean path from we first did confirm the biology, then see if, because you cannot have an effect without confirming the biology. At least so you have to really make sure what you see has an explanation.
Well, thanks a lot for this great discussion, Dominique. All your very hard work and discussion. I see there's a lot of excitement on this new randomized trial that you just published in Nature Aging. A lot of scientists, top scientists. I saw Professor Eric Topol from close to you, where you are in San Diego, talk about how this was unprecedented almost for supplements to do this kind of a rigorous trial. Any last thoughts on the data, on longevity and immune aging? And with that, after that, we'll say thanks to the audience for joining in.
It's obviously a very growing field, an exciting field, and I just encourage people to open the paper. You don't have to be a scientist. Just look at the introduction and discussion. I think we tried very, very strongly to explain it on a basic level and discuss our findings rigorously. There's weaknesses to the study, as every study has, but it's important to keep those in mind. I think that really helps even non-experts to be part of the discussion, because that also helps us to design the next studies and to see what's important to people. So have a look there. There's good summaries out there also by Nature Aging. They published a research briefing, which is a lay summary of the findings. Just be involved in science, I can just say to everybody, and in these times, it's worth it. It's rewarding.
Fantastic. Thanks so much, Dominique. And thanks to you, folks who joined to listen to our AMA. Feel free to post questions. This will be posted on YouTube. So you can always plug in your questions, and either Dominique or I will get back to you. And look up our research. Go to www.timeline.com and look up all the research we've been doing for almost 10 plus years in the clinical space, and even for 15 plus years on the molecule. It's pretty unprecedented. So thank you. We'll post the reference. I see there's a last question. What's the reference for the paper? We'll put the reference in the discussion and in the show notes of this AMA when we post it. Thanks, everybody. Have a great weekend. Thanks, Dominique.
Thank you. Bye.